itself. I think this first slide here illustrates very much what we're doing. We are heavily focusing on commercializing the Clostridia process for N-butanol production. What we see here is an American plant in central Minnesota, which we have agreed to buy up later this year and retrofit to butanol production. The platform we're using is the Clostridia process. It was developed in Manchester by Adaptive Laboratory Evolution 100 years ago, so nothing new really on that part of it. It was then scaled up mainly in Canada, and it got commercialized in 1919 after the First World War, and then it's been running until the 80s. So it has a good 70 years of proof of, more than proof of concept. It was the way to produce N-butanol until oil became too cheap. Now oil is going up, and we are hopefully back in business. And here you see some of the production plant that have both from South Africa and British acetones in Toronto. Why are we doing this? We have changed a little bit on our focus over the last few years, and it's not really broadcasting ourselves as a biofuel company. We are a company which are focusing on producing biochemicals, so commodity chemicals, especially butanol, but we also do have byproducts from the process in form of acetone. Biobutanol or N-butanol is a commodity product which goes into a number of different products, both in the cosmetic areas, but also in paints, solvents, lacquers, butyl acetate, butyl acrylate, butyl glycol, and it's produced in relatively large amount, which means that it's relatively cheap, so it's around $1,600 per ton, which means that there is significantly cost implication there. You can't have an expensive process or use an expensive feedstock. Our focus has been on, not really on building a demonstration plant, even though that's happening now, but first of all, to prove that it actually works in scale, and we've been working heavily with China, where the Chinese government decided to build some plant and to get up production, and then they figured out that we also need to increase the cost of starch for the farmers. So they got all shut down, and we've been working with them, introducing them to work on lignocellulosic hydrolysate, corn cob, and corn stover hydrolysates, with all the problems that's associated with that. And that has helped us tremendously with increased funding, and we have produced butanol there at 4,000 cubic meter scale, so quite big, and has produced, I guess, around 400 tons of N-butanol, which has gone into to start up the market, selling it to chemical companies, which are then producing butyl acetate, butyl acrylate, and so on. So we have off-take agreement, because when you start up a plant and retrofitting a plant, you need to have off-take agreement, otherwise you can't get the financings in there. The other really important thing with the financing in these situations is return of investment, which is something when I was working in academia, was not really high on the list. But generally, if your process, if you don't have return on investment within 4 years, you don't get funding from it. You definitely do not get funding from a capital venture arm. So it's all about cost, and it's about getting the plant paid back really, really fast. So this is, as I said, what we've done in China. We produced enough to sell it to chemical companies, and work heavily on getting a off-take agreement. I think this is in principle a little bit the most important overall slide of it, and I think there's three things which drives this economics. There is the concentration of your product in the broth, there's the yield on the feedstock you're having there, and you really need to think about the yield on the feedstock, not necessarily the sugars in there, because the more you can use of the feedstock, not only the sugars, the more your principle, the cheaper your overall feedstock it is. And then the last part of it is productivity. And out of these ones, I think the productivity is by far the most important one here, especially in this process where this history have already shown that we got good yields in there. So a little bit the background for that is that if you want to start up production, retrofitting an ethanol plant is by far the best option for return on investment. There's no way you're going to start up a plant here in the UK anytime soon with a 250 million pound investment, because you're not going to get it back within 4 years. But retrofitting ethanol plants, especially in the States, is a much better option, and because they're relatively cheap because of the ethanol market there. So we have been working on that, and one key important thing is here that when you both retrofitting it, but you're also building new plants, you normally have a fixed fermentation volume. And it's all about pushing as much product through that fermentation volume as possible in the shortest amount of time. Clostridium strains, they produce solvents in high yields, they have an extended substrate range, and they're relatively good inhibitor tolerance. So that's actually the principal background here. I think the background, one of these sort of targets, we're also looking at it and associated with this volumetric productivity. There's two things there in volumetric productivity. There is specific biomass, specific productivities, and then there's how much biomass or active biomass you got in the fermentation. And that is really the two crucial things right there. Clostridia, they are known to go into differentiation, forming spores, and then they're shut down for various reasons. What is really important here is that membrane stress is that one of the key things leading to sporulation. Can we avoid biomass inactivation? Also, what we often see that you never really get fantastically high biomass concentration in Clostridia processes, not like yeast where you can go up to a wet volume of 400-500 gram per liter broth. This is much lower, and normally in the standard batch one, you don't get above 8-10 gram per liter of cell dry weight. In more advanced mode, where you retain the biomass, you can get up to higher. So that's how can we actually do this ones here? A number of these Clostridia, they do use chlorine sensing. Is that something we can play with? How does it sense the environment? That has to be via the plasma membrane. Can we somehow disrupt that so it grows up to higher biomass concentrations? Butanol is again the product there. It's not much known about how it gets out of the cell. It is hydrophobic, so it will be soluble in the plasma membrane. We'll get back to that later. But is that the only way it gets out there? Like you got other products also. For example, it was long time known that you didn't really how the water get out of the cells, but now there's a number of aquaporins which are facilitating that transport. Is the facilitators also for butanol transport? We don't know. And is diffusion limited, or is it something else? It's not known at all. The other thing is that with Clostridia, they don't really grow well in high substrate concentrations, which means that you normally need to have some kind of fed batch process where you're actually diluting out some of your product, because you're starting with lower concentrations. Yeast, for example, can start at higher ones. What can we actually do there? Why doesn't Clostridia grow at high substrate concentrations? How does it sense the environment and relay that information into the cell? Is it purely osmotic, or is there something else? It's not really known, but it is a mechanism where it's transported. The information is transported over the plasma membrane. Concentration part of it is also somewhat important. Butanol is toxic to microbial cells. It doesn't matter which microbial cell you're having. It could be yeast, it could be E. coli, it could be Clostridia, it could be Lactobacillus, you name it. It is pretty toxic, and you don't normally get viability over a few percent of butanol, no matter what you are saying, and you definitely will not be able to develop a cell which are having a 10-15% tolerance to butanol. Butanol is hydrophobic. It has a log p value with distribution coefficient between octanol and water, decanol and water, I can't remember, one of those two ones, 0.88. Ethanol is 0.19, so there's a tenfold higher solubility within lipid compartments, plasma membranes. How does that influence the system? Unfortunately, also butanol has a high boiling point, so you can't just evaporate it off like you could in principle theoretically with ethanol. So given all this background, butanol tolerance is still really, really important. What it means is that the process you're running in it, you need to have some kind of in situ butanol removal in it. That is an engineering solution to it. And it always costs money and to have to remove butanol in there. So the question is, how can we actually remove this butanol? As I say, you can talk about engineering solution on it, but one thing is clear: by having a higher tolerance to butanol, you'll be able to operate the process at a higher butanol rate, which means that whatever process you have to remove the butanol is going to be cheaper. And that is really important when you're going to make a product which sells for $1,600 per ton. So there's a number of, I think, characteristics here which we are interested in, that is understanding how butanol is toxic. Is it the plasma membranes it disturbs? Is it something else? Is it protein folding? But clearly that it could be a mixture of those two. Can we change the plasma membrane composition, make it more resistant to butanol tolerance? It again comes back to how butanol is toxic. Is it specific membrane proteins which are really, really sensitive to butanol? There's a number of so ATP synthase is a really, really important energy generating factor, and there's a number of redox carriers and proton exporters to generate this proton motive force over the plasma membrane, which is really important in the energetics of the Clostridia cells. How are they actually impacted by this butanol concentration and all this sort of hydrophobic material lying around? How is it transported out of the cell? Is it diffusion? Is it facilitated diffusion? Which ones is it? Can we upregulate the carriers of butanol out of the cells? Again, I sort of talked a little bit about how does butanol influence proton leakage across the membrane, and in that way the whole energy characteristics of the cell, which are influencing the yield ones. And also you can just say like, you can modify the plasma membrane. Maybe you can have some cyclopropanes derived compounds in there. Maybe you could think about, can we change it more drastically, like hopanoids or somehow that, which is like a cholesterol type ones. Will they improve the process? So there's a number of things trying to understand why is butanol toxic, and how can we change the membrane composition, both in the fatty acids, the type of fatty acids, the type of lipids there, and also the proteins in the plasma membrane. And the last part of this tree characteristic is yield. I think yield is still really, really important, because sugar cost is a major cost factor for butanol production. And for a corn butanol plant, is around 70% of the operating cost is sugar. So if you can add something to that, then that is valuable. Clearly not as valuable as the productivity, because if you can dump twice as much product out of the same plant, you earn twice as much money. While this is a differential one. So the maximum yield of butanol in any kind of strain on pure sugar is 0.41 gram per butanol per gram sugar. It doesn't matter which strain you're using, whether it is a yeast or E. coli. There's no way around it. It's a chemical defined one there. But there's other ways a little bit to get around it. So this is constrained by its redox metabolism and the redox balance. I'll come back to that a little bit later. The other little bit of background here is that when you take these strains, the most cost-effective way of generating ATP to deal with some butanol stress or whatever it needs sort of to grow and maintain itself is acetate production. You don't want to have acetate, produce acetate. It's a low-value compound, and you don't want that. So the targets here. So there's several ones coming out of this sort of discussions or thought processes that how can we minimize ATP consumption used for substrate uptake. So that could be looking at different kind of transporters, transporting up oligomeric material, so it's at lower cost when it gets in into the cell. Also, what's the cost of products getting out of it? Then there's utilizing additional substrate in the feedstocks. So that could be acetates and that kind of things. A lot of lignocellulosic hydrolysis has acetate in there. If you can use that one up, then your yield of sugar goes up because you got extra carbon, and there's a number of other substrates there, which is really relevant. Yep. Also, what's say like we're really focusing on the commercialization of it and getting that up and running, and we need to that by our capital venture and our investors in there. So we don't really have too much basic research within the company, but we do sort of would like to do a little bit more rational improvement of the strains. What we don't really know is how and what genes, proteins are responsible for uptake of additional substrates. Often you know sort of generally about your feedstock, what substrates in there, but also what is a sugar. So people who's been working with hydrolysate and sugars, you always know that a given sugar is not just one thing, there's different things there. You got the furanose form, you got the pyranose form, you got the alpha, and you got the beta, and then equilibrium. It's not always a really fast equilibrium. You see that also from some of the gene context there. So like, what is actually coming out of the feedstocks? Can we induce sort of the uptake of some of these other forms of the sugars? Will that improve the process? Understanding that, generally understanding which genes in the strains, the procedure change you're working with, which are responsible for uptake of these additional substrates in there. Minimize the uptake of fermentation inhibitors. So with the idea that could be lignin-derived aromatics or fulfurol. If they're not taken up, then hopefully they won't be toxic. So can we knock that out? Or on the other way, you can look at it. Can we detoxify them so fast inside the cell that we are not losing energy and we can maybe actually turn them into a substrate instead? That would be another way of looking around. The other thing is that, as I say, like the maximum yield of butanol from sugar, that is restricted by the redox balance. So if you pump in more redox power into the system, you should be able to increase that kind of yield. Again, there's potential options there about hydrogenase. They do have hydrogenases in there. They are rumored to be reversible, that kind of things. Can we use that actually to directly drive carbon-free reduction equivalents into the system, so we can increase the butanol yield on sugar and thereby increase the economics of the process? So that's another membrane area which I think is important, could be important for us. There's also general topics. So we have sequenced a number of strains, and you sort of look through it. You try to build also metabolic model to do a little bit of rational or logical argumentation for what you should do. And you often see like, what is it doing here? And it's very well known that also these strains, they use the RNF system for getting the most energy out of its substrates. There's a linkage between NADPH and NADH and ferredoxines that can be used to pump out protons out of the cell, which then can be used to generate ATP of it. But then there's the other thing also. A number of these strains, they got this NADH dehydrogenase 1 complex, which is totally a mystery for me. It's in all my knowledge, it's an aerobic system. These one strains, they are complete anaerobes. Some of them tolerate a little bit of oxygen. So what is it doing there? It's known. Okay, these ones, they do pump out protons out of the cell, and then the electrons are released to the plasma membrane. So clearly it has some kind of effect there, and it takes up reduction equivalents. Like what is happening there? It's not known what the the electron donors is. Is it NADH, NADPH? Is it something else with a pumping in there? It's not known which plasma membrane electron carrier it goes into. Some of them they seem to have something like a minikinome type biosynthetic pathway, but it's definitely by far not complete. And it is at all not known what is the terminal electron acceptor. So there's a whole thing of membrane function there, which is totally not known. And I think it's not only potentially important for us, and I say potentially there because there's no guarantee it's going to be important. If there's guarantee it's going to be important, then that we get something positive, then we'll probably fund it ourselves. But at least what we're trying to look at, trying to see, is there something in it, and is there something we should have a follow-up project on where we can sort of try to improve our process. But then, of course, also you have to think about Clostridia. They're very diverse organisms, and there is a number of other Clostridia which has interest from more medical sides of it. And of course, generating sort of the general knowledge about Clostridia membranes with membrane function might have some consequences also for other things. And I think with that, I will stop.
Yes, excellent.
Thanks.
Thank you very much.